Okay, so previously we have looked at the primary structure of proteins. We are now going to continue with the protein structure, and today we are going to study about the secondary structure of proteins. But before moving forward, we are going to quickly take a look at some important pieces of information pertaining to the protein structure and the peptide bond. This is a diagrammatic representation showing the peptide bond. Here you see this is the alpha carbon of one amino acid. This is the carboxyl group. This is another amino acid, the alpha carbon. Its carboxyl group is not shown, and this is the amino group of this second amino acid. So this is one amino acid, and this is the other amino acid. So the carboxyl of this amino acid forms a peptide bond with the amino of this amino acid. So this bond between the carboxyl and the amino group, that is the amide linkage, it is the peptide bond. The peptide bond has a partial double bond characteristic. These electrons between the carbon and oxygen atom, they are delocalized. So these electrons, they could be transferred between carbon and nitrogen as shown in this diagram here. So because of this delocalization, the peptide bond has a partial double bond characteristic. Because of this partial double bond characteristic, the peptide bond is planar. Rotation is not possible across the peptide bond. In this figure, this rectangle you see, that is representing the plane of the peptide bond. Other bonds like alpha carbon and carboxyl, alpha carbon and the side chain, these bonds are single covalent bond and rotation is possible across them. But the peptide bond, because of the delocalization of these electrons, it has a partial double bond characteristic, and hence there is no rotation across the peptide bond. It is a rigid planar bond. This is a diagrammatic representation showing the peptide bonds in a given protein. Here you see this is a fully extended conformation of the protein. Actually, when proteins are formed, the structure of protein is not like this. This is the structure of the protein opened. So you see that this is a peptide bond represented by this shaded square here, and all of these shaded regions, they represent the peptide bond. These are the side chains of these amino acids. The side chains, they interact with one another. Also, they interact with the solvent as well. So because of these interactions, the protein is going to fold into a particular structure. So this part, the peptide bond linked to the alpha carbon, then again the peptide bond linked to the alpha carbon, the peptide bond linked to the alpha carbon. So the peptide bond associated with the alpha carbon, this whole region is called as the backbone of the protein, sometimes called as the polypeptide backbone. And these are the side chains which are projecting on the sides here. These are the side chains of these amino acids. So the backbone of the amino acids as well as the side chains of amino acids, they do interact with one another and also the solvent as well. So therefore, these interactions, they lead to a particular pattern of folding of the protein. This is a diagrammatic representation of the polypeptide when all of the peptide bonds, they are fully extended. Actually, the structure of proteins is not like this. This is a diagrammatic representation showing two peptide bonds here. So you see this is a peptide bond. This is a carboxyl group of one amino acid. This is the amino group of another amino acid. So this is the peptide bond shown by this plane here. The alpha carbon is represented of here. This alpha carbon is linked to the amino group, and this amino group is in turn associated with the carboxyl of the next amino acid. So this is the next peptide bond. The plane is showing the peptide bond here. So across this bond axis, alpha carbon and the carboxyl, and alpha carbon and the amino, rotation is possible across these two bond axes. We have just seen that the peptide bond is planar, it is rigid bond. So therefore rotation is not possible across the peptide bond here as well as here. However, these bonds between the alpha carbon and the carboxyl and the alpha carbon and the amino, they can undergo rotation. So the bond between alpha carbon and the carboxyl is called as the psi bond, and the bond between the alpha carbon and the amino is called as the phi bond. So across these bonds, rotation is possible. Sometimes the peptide bond is also called as the omega bond, and rotation is not possible across this bond. So rotation is only possible across the phi and the psi bonds. Now, since rotation is possible across these bonds, so therefore these groups, they can rotate clockwise or anti-clockwise. Same goes, rotation is possible across this bond, so this peptide bond can rotate clockwise or anti-clockwise. However, since there are many amino acids and several of the amino acids, they have bulky side chains as well, so only some conformations are possible. This is a diagrammatic representation showing the phenomenon. Here you see this is a peptide bond. The amino group and the carboxyl group, they're linked through a peptide bond here. This is the alpha carbon, and this is the other peptide bond shown here, carboxyl and amino. So this is the other peptide bond. So although the phi and the psi angles, rotation is possible across these bonds, however, because of steric hindrances and clashes, only certain conformations of these amino acids are possible. Because if they come very close to each other, they will collide, the groups will collide with one another, and therefore the structure will become destabilized. So therefore only certain possible conformations are permissible. This property is utilized in terms of this Ramachandran plot. Ramachandran was the name of the person who devised this mechanism. The plot is actually between the phi angles on the x-axis and the psi angles on the y-axis. So these bond angles are determined. One of the ways to determine the bond angles is X-ray crystallography, where the proteins are crystallized and then X-rays are incident on them. And by solving the structure of those atoms making up that protein, the phi and the psi bond angles, they could be deduced. So this plot, what it shows is that there are only certain permissible conformations in which amino acids can exist when they are present in a given secondary structure. For example, we know about one of the secondary structures which are called as the helices. Those amino acids which form the alpha helix, they have a certain range of phi and psi angles, and they typically fall within this region. Those amino acids which form the beta sheet structures, which is another secondary structure element, so their bond angles are such that they are going to fall within this region. So this observation was made by observing the phi and psi angles of many different proteins where amino acids were present either in alpha helices or in beta sheets. So when their phi and psi angles were plotted, it was found that for alpha helices, those amino acids are going to be localizing in somewhere in this region. And those amino acids which are forming the beta sheets, their phi and psi bond angles are going to be localized into this particular region. The Ramachandran plot is typically important with respect to the prediction of secondary structure elements. Let us say if we know about the phi and psi angles of a given amino acid or a certain region of a protein, then we can simply produce a plot of the phi and psi angles and see in which region of the plot those amino acids are going to be present. If the amino acids are going to be concentrated in this region, that means that particular part of the protein is going to form an alpha helix. Or if those amino acids are going to, their phi and psi angles are such that they're going to localize in this particular part of the plot, that means they're going to form beta sheet structures. This is a Ramachandran plot which is representing the various conformations of glycines. Now we know that a glycine is the simplest amino acid and it contains a hydrogen atom as its side chain. Now hydrogen atom is a very small species and it occupies a very little volume. So therefore glycine, it can exist in many possible configurations. This is also represented by this graph, by this plot here. So you can see that these are the regions where glycine corresponding to the phi and psi angles of glycine. In all these possible configurations, the glycine is present in proteins. So this is the simplest amino acid with the smallest side chain, which is a hydrogen atom. So therefore, there are least steric clashes and hindrances. Because of this, the diversity of phi and psi angles into which glycine can fit is much more as compared to any other amino acid. So glycine is much more flexible as compared to any other given amino acid, simply because of the reason that its side chain is very small, occupies a very little volume, and therefore could be accommodated in many possible different bond angles. So amino acids can exist in several possible permissible conformations, and because of this little conformation flexibility, the amino acids in the backbone of the protein can assume several types of secondary structure elements. So the amino acids, the backbone and the side chain, they do interact with one another, and they give rise to the overall architecture of the protein structure. Now we are going to quickly look at some of the important types of secondary structure elements. The first one in this regard are called as the helical regions of the proteins, or more famously called as the helix. Helix could be formed in many different possible ways, but the most common helices found in proteins are called as the alpha helices. A helix is a region in which the amino acids are organized in a spiral conformation, as you can see here. This structure is produced, the alpha helix is a structure which is produced when the amino nitrogen of one amino acid interacts with the carboxyl, with the oxygen of a carboxyl of the fourth amino acid. So this is one amino acid here, this is the second amino acid, the third one, and this one is the fourth one. So this is the carboxyl of the fourth one. This is the carbon, that's the oxygen. So this oxygen, being electronegative, interacts with the hydrogen atom of this amino group. So the amino group of one amino acid interacts with the carboxyl group of the fourth amino acid. So this is going to happen for all of these amino acids. So that means the second amino acid, its amino group will interact with a carboxyl of the fifth amino acid. The amino of the third amino acid is going to interact with the carboxyl of the sixth amino acid. So in this way, all of these amino acids, they are going to be linked to one another by means of these hydrogen bonds. And these hydrogen bonds are formed between the hydrogen of the amino group and the oxygen of the carboxyl group. So in this way, the polypeptide backbone is going to be forming a helical structure. When such a bond, a hydrogen bond, is formed between the first and the fourth, every first and fourth amino acids, this structure is called as the alpha helix. Proteins contain many other different types of helices as well. So if the bond formation is between the first and second amino acid, it's called as a 2.2 ribbon. If it's between first and third amino acid, that is called as a 310 helix. If it's between first and fourth amino acid, that is called as alpha helix. And if the hydrogen bond formation is between the first and the fifth amino acid, that is called as a pi helix. Alpha helices are the most predominant type of helices found in the proteins. However, certain parts of the protein, they also contain other types of helices as well. Okay, so the next important type of secondary structure element are called as the sheets, sometimes also called as the beta sheets. The sheets are formed when a given region of a polypeptide chain forms hydrogen bonds with the adjacent region of the polypeptide chain. So here you can see that this is one segment, one region of the protein, and this is another region of the same protein. These two regions, they lie close to each other, and they are going to form hydrogen bonds with one another. So this is one portion of the protein, and this is the amino group of this portion. This is a hydrogen atom. It forms a hydrogen bond with a carboxyl of the other segment of the protein. And this is the next amino acid of the first segment, and it forms a hydrogen bond with the second amino acid of the second segment. So that's the third, fourth, fifth, sixth, and so on. So these consecutive amino acids are present on this backbone of this particular part of the protein are going to form hydrogen bonds with another series of amino acids on some other segment of the protein. Such structures, they're called as the sheets. In this first illustration, you can see that the these two regions, they're projecting in the opposite direction. That is, this is the N terminus of this segment, and here is the C terminus of this segment. And for the other segment, the N terminus lies on right side, and the C terminus lies on the the N terminus lies on the left side, and the C terminus lies on the right side. So these two sheets, they are running in the opposite direction as compared to one another. So these are called as the anti-parallel beta sheets. The other type of sheets are called as parallel beta sheets, and in case of parallel beta sheets, the pattern of hydrogen bonding is similar, except the fact that both of the segments of the polypeptide are running in the same direction. They're running parallel to each other. So such secondary structure elements are called as beta sheets. Sometimes it happens that many sheets, they form overlapping regions. They are called as beta pleated sheets. Other important secondary structure elements include turns, loops, and coils. Turns are the regions which are typically made up of around 4 amino acids, and turns, they produce a sharp kink, a sharp bend in the polypeptide backbone. So you can see that this is a polypeptide backbone running here, and then these 4 amino acids, they produce uh, a kink, a bend in the structure of this protein. This is another type of a turn shown here. So this is one secondary structure element. So you can see this is one sheet, there's a turn, and then this is another sheet. So a turn is a short segment of the polypeptide backbone which produces a kink, a bend in that polypeptide backbone. That is called as a turn. Loops are generally longer structures as compared to turns, and they also result in flexibility of the polypeptide backbone. Coils are random regions which do not have any secondary structure elements in them, and they are sometimes called as the random regions, but actually they are not completely random. Coils are generally the unstructured regions which cannot be classified into helices, sheets, or turns. Those regions of the protein are called as the coils. So all these are secondary structure elements. A secondary structure is a local region of the protein. These secondary structure elements, they do interact with one another, and they give rise to the overall architecture of a protein. For example, here in this particular example, this is a diagrammatic representation of a structure of a protein. So these blue colored regions are representing the helices. So you can see that helices are distributed on the periphery of this protein. In the central region of the protein, it contains sheets in it. The sheets are represented by green colored arrows. And these portions, which are shown in orange color, these are the coils and loops. This is another view of the same protein. Some proteins contain largely helices in them. Some contain exclusively helices in them. Other proteins, they largely can contain sheets in them, but a variety of protein contain both of these secondary structure elements. This is called as the tertiary structure of a protein. So all these secondary structure elements, when they are interacting with one another and also interacting with the solvent around them, they fold into a particular three-dimensional overall architecture of the protein. This overall three-dimensional structure of the protein is called as the tertiary structure of that protein. Alright, so now we are going to look at some important structural features of various proteins. The first one in this regard is keratin. Keratin is a structural protein. It is a fibrous protein. Fibrous proteins are those which form fiber-like filaments. Keratin is found in the hair, nails, horns, and the hooves of the animal. Keratin is also associated with the skin, and it has a protective function. This is a diagrammatic representation showing the organization of keratin protein. The protein is a fibrous protein, that is, it forms fibers. So the two molecules of the protein, they form a dimer. They are wound over each other in form of a helix. So they form a dimer. And then many of these dimers of the protein, they arrange with one another and they form a higher order structure which is called as a protofilament. And then many such protofilaments, they associate with one another and they give rise to this structure which is called as the microfibril. And this is one of the microfibrils which is shown here. Many such microfibrils, they form a macrofibril. And a lot of these macrofibrils, they make, along with associated cells, they make the entire hair. So this is the structural organization of keratin found in hair. So keratin is a helix of helices. It's a helical protein in which the monomers are arranged in form of helices, and then these helices are further organized into higher order helical structures. Some people have curly hair, others they have straight hair. So the reason why the architecture of hair is different is because of the pattern of disulfide bonds. This is a diagrammatic representation showing the process of curling of hair. So see that these are the fibers and they have disulfide bonds between them. So a reducing agent is added. Reducing agent is going to reduce these disulfide bonds and produce sulfhydryl groups. After reduction, the hair is curled, which alters the shape of the individual protein fibers, and then an oxidizing agent is added. Upon addition of the oxidizing agent, the adjacent sulfhydryl groups, they are going to form disulfides. Now you can see that the pattern of disulfides initially and at the end of the process is very different. So in this way, straight hair, which have a certain arrangement of disulfide bonds, could be converted into curls with an altered pattern of disulfide bonds. The next important structural protein is called as collagen. Collagen is a structural protein. Again, it's a fibrous protein which is found in different types of body matrices. It is associated with the bones. It is found in the gums and several other body matrices. Collagen is important because it provides physical strength to different body matrices. Collagen has derivatives of the standard amino acids. It contains hydroxyproline and hydroxylysine. So this hydroxylation is important for the structural integrity of collagen. Collagen is a triple helix in which three chains, they are arranged in form of a helical fashion here, as depicted in this diagram. This hydroxylation of the amino acids is important for a typical pattern of hydrogen bonding, as represented in this diagram. So here the individual fibers are shown extended, separated from each other, and they are interacting with one another through hydrogen bonds. If hydroxylation of these amino acids do not take place, then proper folding and arrangement of this protein does not take place. That decreases the mechanical strength. Vitamin C, ascorbic acid, is a cofactor for carrying out the hydroxylation of these amino acids. You would remember from high school biology that a deficiency of vitamin C causes scurvy. Scurvy is a disease in which bleeding through the gums takes place. So when people have a deficiency of vitamin C, they are unable to carry out hydroxylation of these amino acids properly. And when hydroxylation of proline and lysine does not take place properly, then these fibers cannot fold into the proper structure of the protein with greater mechanical strength. So as a result of improper or no hydroxylation, the protein fibers become weak. As a result, they could be easily damaged by pressure or external force, and therefore that results in bleeding of the gums. So the deficiency of vitamin C causes scurvy, in which bleeding takes place from the gums, and the reason of bleeding is decrease in the strength of collagen fibers, which is because of improper hydroxylation, because vitamin C is a cofactor for the hydroxylation of proline and lysine. This is an electron micrograph showing the structure of the collagen fibers. Moreover, the collagen fibers are also covalently cross-linked to one another, as shown in these regions. So the proteins discussed so far are examples of fibrous proteins which form long fiber-like filaments. The next type of proteins are called as the globular proteins. Globular proteins are those which have nearly spherical structures, as opposed to the fibrous proteins which have elongated structures. Hemoglobin and myoglobin are two common examples of globular proteins. Hemoglobin is an oxygen transporting protein found in the red blood cells, and myoglobin is a protein found in the muscles which carries out the same function. So this is a diagrammatic representation of a globular protein. So the globular proteins are folded into a structure such that they are going to form a spherical organization of the structure. Some globular proteins may be rich in alpha helices. For example, hemoglobin and myoglobin, they both contain a large number of helices and they do not have any sheets in them. Some globular proteins, on the other hand, they are rich in beta sheets. For example, this protein represented here contains many sheets in it. The sheets are represented by all these colored arrows. However, many proteins, they contain a mixture of helices as well as sheets, which give rise to the tertiary structure of the protein. Now we are going to quickly look at some of the important factors which affect protein stability and therefore its folding into the three-dimensional structure. One such important force is the electrostatic interaction. This diagram is representing the surface of a protein, and the charged amino acids are represented in red and blue color. So you can see that the surface of the protein mainly contains the charged amino acids. The charged amino acids, they have electrostatic interactions between them. For example, this is a representation of a lysine and a glutamic acid. So a glutamate contains carboxyl and lysine contains an amino group. So amino is positively charged and the carboxyl is negatively charged. Similarly, the aspartate and arginine is positively charged and aspartate is negatively charged. So these oppositely charged regions of the amino acids, they have electrostatic interactive forces with one another. So this is one important type of interaction which is found in proteins. Most of the polar and the charged amino acids, they are present on the surface of the protein. The other type of interaction are the dipole interactions. There are many different types of groups which are themselves not polar, but the constituent atoms, they have a difference of electronegativity. For example, this is a carbonyl in which the oxygen is more electronegative atom and the carbon is less electronegative atom. Since oxygen is more electronegative, so it will withdraw the shared pair of electrons towards itself. As a result of which, this oxygen will become partly negative and a carbon will become partially positive. So this is a dipole, and this dipole can interact with other dipoles. So there could be a dipole which can induce dipole in other molecules as well. So collectively, these dipole interactions are also important for the stability of the structure of proteins. The next important ones are the hydrogen bonds. Hydrogen bonds are formed between a hydrogen atom which is linked to an electronegative atom. So this hydrogen atom, which is bound to an electronegative atom, is attracted by another electronegative atom in its vicinity. This is called as a hydrogen bond. Hydrogen bonds are also an important chemical force in stabilizing the structure of proteins. Another very important interaction is the hydrophobic interaction. Hydrophobic interaction takes place when non-polar substances or hydrophobic substances are present in a polar environment. Water is a polar environment present in the cell. So these amino acids which contain hydrophobic side chains, for example, the aromatic amino acids or the aliphatic amino acids, they are non-polar. So these non-polar hydrophobic amino acids, when they're present in an aqueous environment, they're unable to interact with water. As a result, they are going to interact with one another. So this interaction is called as hydrophobic interaction. Mostly the hydrophobic amino acids are going to lie, they're going to be buried inside the interior of the protein structure where they are protected and reside away from water. They're not exposed to water. Mostly the polar and the charged amino acids, they're going to lie on the outside of the protein structure close to the surface. And the non-polar and the hydrophobic amino acids, they're buried inside the three-dimensional structure of a protein. This plot is a hydropathy plot, and this is representing the hydropathic index on the y-axis. Uh, Increase on the y-axis means more hydrophobicity. Decrease on the y-axis represents more hydrophilicity. On the x-axis is the number of amino acids. So you can see that these are the portions. The peaks are the portions which contain amino acids which are more hydrophobic. So these portions which are forming the peak are expected to be buried deep inside the protein core. Another important type of interaction is a covalent bond. The previous ones which we have studied, they are all non-covalent interactions. Ionic interactions, van der Waals interactions, dipole interactions, hydrophobic interaction, hydrogen bonds, they all are non-covalent bonds. Disulfide bond, however, is a covalent bond, and we've talked about this earlier. The disulfide bonds are formed between cysteine amino acids whose sulfhydryl groups are present in proximity of each other in space, and they can undergo oxidation to produce a disulfide bond. This is a diagrammatic representation showing the structure of the enzyme ribonuclease A. The ribonuclease A contains four disulfide bonds in it. And this protein has a very unique feature that when these disulfides are reduced, the protein undergoes a reduced denatured structure. But when the denatured protein is again exposed to oxidizing conditions, it again falls back into this original three-dimensional structure by reforming these disulfide bonds. The last level of structural organization in proteins is called as quaternary structure. Quaternary structure exists for only those proteins which have multiple subunits in them. Such proteins are called as oligomeric proteins. Those proteins which are made up of only one polypeptide chain, they're called as monomeric, and their structural organization is the tertiary structure. However, when there are more than one polypeptide chains in a given protein, for example, hemoglobin contains four polypeptide chains in it. So each of the polypeptide chain has its own given tertiary structure. And then these polypeptide chains, they interact with one another through specific interactions, and now they give rise to another higher level organization of the entire protein structure. So, for example, in case of hemoglobin, there are these four polypeptide chains which interact with one another in a specific particular manner, and they always engage with each other in this particular structural manner. So, this structural organization where several polypeptides having their own typical tertiary structures, they interact with one another and they form a higher order structure of the protein structure that is called as the quaternary structure of these proteins. Quaternary structure exists only for oligomeric proteins, the ones which have more than one polypeptide chains in them. Many proteins form complexes, complexes with one another or complexes with RNA as well. For example, ribosome is a complex of many different proteins and RNA molecules. In this representation, you can see the viral capsid. The viral capsid is going to form an outer covering in which the nucleic acid of the RNA is packed. So these viral capsids, these are formed by interaction of many different subunits of the protein, and they give rise to this entire structure of the capsid. Um, the complexes of the electron transport chain, for example, are also made up of many different types of proteins. Pyruvate dehydrogenase complex is a complex of enzymes which is important for the conversion of pyruvic acid into acetyl CoA. So this is a process. This is a chemical reaction which is important to feed acetyl CoA for the Krebs cycle. So pyruvate dehydrogenase complex is also a multi-subunit complex. So many proteins and enzymes, they fold or pack into these large ordered structures. So with this, we come to an end to this topic about the structural organization of proteins.